Okay.
Yes, sir.
Today our topic is disaccharides.
Yes, sir.
And what are disaccharides? As we know, the disaccharides are the carbohydrates
Yes,
which
sir.
are formed by the combination of
Two mon
two monosaccharides,
two monosaccharides.
right?
Yes.
Or two simpler sugars.
Yes, sir.
All right. So, for example, we have an example of maltose.
Yes, sir.
Maltose is actually it is the combination of two glucose molecules.
Yes, sir.
All right.
Yes, sir.
So yesterday we have discussed about the how the because glycosidic bond is always between the two glucose in disaccharides, and then it will be in polysaccharides. Definitely, it is not the part of monosaccharides because monosaccharides have only one molecule of sugar, that may be glucose or galactose, and that we have discussed a few examples in
Yes, sir.
monosaccharides.
Yes, sir.
So in this case, for example, you have two glucose. We can draw, and you must know about how to draw these structures. You must be familiar with these structures as well. So because you can only differentiate these structures that, for example, and there's a one structure here. On this side, you have another structure like this,
Yes, sir.
like like this. For example, you have two uh, these glucose molecules which are present there. So in this case that we have discussed even yesterday as well. For example, you can also number them one, two, three, four, five, and this will be six because carbon number six is outside.
Yes,
On the
sir.
other hand, you will here is four. Okay. On the other side, you can also give the number. This is one. This is two. This is three. Here is four. You have five, and then you will have six because carbon number six is again outside.
Yes, sir.
So in this case, we can discuss that. For example, here you have OH, and in a similar way, you have another OH on this side. And definitely on this carbon you have hydrogen, and this carbon you have one hydrogen. So
Yes.
as we have discussed yesterday that
Yes, sir.
because of condensation,
Yes, sir.
hydrogen will be from one glucose molecule and hydroxyl is from the other glucose molecule. They will combine with each other and they will produce or synthesize water here. Is
Yes.
it?
Yes.
Okay. So this is a condensation process. Another thing is it is what we call the bond which will be formed here, that will be glycosidic bond. All right.
Yes.
So water will remove here. So what will be here that is remain like that is only oxygen, right? Because hydrogen is removed from here, and you have, for example, you have this oxygen, right? So
Yes.
now this oxygen is only there. So you will have a structure like
Yes,
this,
sir.
right? So water is removed from here, so only oxygen is remained. So that oxygen is present between these two molecules. So this is what, and we can give its number 1,4 glycosidic linkage. I call it 1,4 because
The
uh, because here in this bond, carbon number 1 and carbon number 4 is involved.
yes, sir.
All right. So these both are involved here. So this is how you will get this maltose,
Yes, sir.
right? So
Yes,
you
sir.
must keep in mind that which carbons are involved. So remember, in this case, in maltose, uh, carbon number 5 or carbon number 2 or some other will not involve, right? Glycosidic bond that needs some symmetry. So that symmetry is only established here in 1 and 4. All
Yeah.
right, in this case. But it will be changed in some other, like we are talking about sucrose.
Yes, sir.
Sucrose. And how sucrose is formed and what are the monosaccharides which are involved here. Monosaccharides which are involved here in case of sucrose.
Glucose and galactose.
No, uh,
Glucose and
fructose, right?
fructose. Yes,
Lactose was actually where it was. We have discussed this. It was involved in sucrose. Also, is it not in the disaccharide? No, it is not involved there. Galactose is actually it is a monosaccharide which is present in lactose, right?
yes, sir. Yes, sir. Yes,
Lactose. In case of lactose,
yes, sir.
in case of sucrose, you will have glucose plus fructose.
Yes, sir.
Sucrose. And in this sucrose, you will have glucose plus fructose. All right.
Yes, sir.
So these are two monosaccharides which are involved here to synthesize a disaccharide that is sucrose. Again, you can draw a structure here, and you should know the difference between, because we have discussed the difference between glucose and fructose, how they are different from each other. Although both are isomers, but they are different because of their structure. As we know
Yes.
that isomers are those molecules which have same molecular formula, but they have different structure formula, right? They have different structure. So in this case, glucose and fructose, they are isomers, but they are different in their structure. So again, the glycosidic bond will form here, but you must keep in mind the symmetry of the bonds between them. It would be, I'm directly writing here, that is 1,2 glycosidic linkage.
Okay, sir.
Here it is 1,2, but in case of in previous case where you have synthesized maltose, in case of maltose it was 1,4
Oh,
glycosidic linkage, but in this
yes, sir.
case it is 1,2 glycosidic linkage, and it would be very easy to understand when you will draw a structure and you will observe there that which carbon is involved in this reaction. For example, we can draw it here. Suppose for example, you have one glucose, as usual the glucose is same here. This, 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 yes, it's 1, it's 2, it's 3, it's 4, it's 5, and it's 6. Okay, one thing you should keep in mind in case of fructose, in case of fructose, you will have some difference. Keep in mind in the structure. Structure is changed like this.
Yes, sir.
Okay, so like
Yes,
this,
sir.
CH2OH. Here you will have another one, CH2OH. I'm writing here. So this is carbon number. From where we should start? Oh, we can start from here. It is carbon number. Uh, hmm, I should write from this side. Uh, it is what? 1, 1, 2 glycosidic linkage. Is it?
सर जी ये
It is one.
जो फ्रक्टोज है
जी जी.
इसमें कार्बन्स कितने हैं?
Carbon 6 हैं, because listen, this is I will change the numbering on that side. I will show you how I've changed how I can change that. Listen, here it is, for example, in case of fructose you have, this is carbon number 1, this is carbon number 2, this is 3, this is 4,
सर
this is 5, and this one is 6. Carbons
यस.
are same because both are isomers of each other, so they have same number of carbons, right? but you can just observe the difference in their structure, because here the structure is changed as compared to glucose, right? I'm
Mm.
just trying to show you that how the numbering is changed here. So in this case, 6 carbons, but they have different number. You can say the number is changed here, right? Their sequence is changed. But in this case, on this side, you have 1, 2, 3, 4, 5. Now the bond will form between what?
Sir, carbon number 2 and
Here
1.
is carbon number 2. This is carbon number 2. And that's another color. Anyway, like this,
Yes,
in
sir.
these two carbons. Here is carbon number 1 of what? Of glucose.
Glucose.
Remember, it is glucose.
Yes, sir.
And carbon number 2 of fructose is involved. Here is fructose. All right. So this is how the glycosidic linkage is formed. So definitely
Yes.
oxygen is there in between them, and this is how the glycosidic linkage is formed. So now it is a sucrose.
Yes, sir.
Sucrose is formed, right? So here you should keep in mind when you will get the question somehow. So you should know about uh, uh, what is the symmetry of these two these two molecules, especially glucose in this case and fructose, and in case of, as we have discussed in case of maltose, so there how the molecules are arranged, and specifically the glycosidic linkage is formed in which two carbon atoms. This
Yeah.
is you should know about. Similarly, you would have another compound. Now you can also draw. Do you have copy pen with you or not? रखा करो copy pen यार.
Mm.
This is how you can, it will be easy for you. So for example, you can draw for lactose. Lactose is galactose plus glucose. So you can draw it, okay? And here
Yes,
you
sir.
will have some difference. Like there you would found that like in this case, you would
One
have
six.
one. It is not 1 6. It is 1 4. 1 4. It is also 1 4, but there you would have difference like uh, here will be oxygen. So this is how you will find, but that is in case of galactose. So you should try this structure as well. You should know how to draw this structure, right?
Yes, sir.
So let's move on to the next point, next discussion, that is polysaccharide. All right.
Yes. Sir, 1 6 कितने होता है?
Polysaccharide. 1 6. Yes, 1 6 is there, but that is in case of polysaccharides. We will discuss this topic as well. Like you would have in case of for example, you would have in case of glycogen, right? It is
Yes, sir.
also present. That is, there you will have 1 4 glycosidic bond as well as 1 6, right?
Okay,
So
sir.
uh, in case of polysaccharides, we will observe this sort of arrangement of the carbon atoms. Like uh, in case of glycogen, you have two types of combination of this glycosidic linkage. One is 1 4, another is 1 6, right?
Sir.
So up till now, in case of disaccharides, we have just studied two things: one 2 glycosidic linkage and 1 4 glycoside.
Yes, sir.
All right. Let's talk about the polysaccharide. So what is polysaccharide? And give me some examples as well. Please, what do you think?
Uh,
Polysaccharide. Polysaccharide. How you will define this term?
sir, a sugar containing different types, more than two types of monomers, or
Yes,
monosaccharides.
monomers of glucose or sugar, whatever you say, right? Right.
Yes, sir.
So we can draw, or we can assume its molecular formula, which we commonly use for polysaccharides. That is CX H2O1, right? It is the general formula that we commonly use for polysaccharides.
Yes, sir.
It is polysaccharides. All right.
Yes, sir.
Okay. So for instance, we can start it from an example, that is starch. Starch. What is starch? Starch is, definitely, it is a stored form of carbohydrates in plants. It is stored form of carbohydrates in plants.
Yes, sir.
All right. Okay. So starch is made up of two components, two macromolecules. Its composition is, it is made up of two macromolecules.
Yes, sir.
All right. One is amylose, another is amylopectin. Amylose and amylopectin. These are two macromolecules by which it is formed. So monomers are the single unit of starches. What would be the single unit here? Because it is polysaccharide, so there must be some single unit, that is glucose.
Yes, sir.
Glucose is the monomer of this starch. By the repetition of glucose, this structure is formed. So let's talk about first amylose, because we have discussed these are two macromolecules. How much time do we have?
सर जी अभी टाइमर नहीं आया.
Okay, time. All right. So amylose. Amylose. What is amylose? Okay. Amylose. It is a macromolecule. It is actually based on long chain of alpha glucose in helical form, right? Helical
Yes, sir.
structure held together by hydrogen bond, right? Not straight. It is not straight. Okay. So all these glucose have 1,4 glycosidic linkage.
Mm.
Remember. So you will have this difference. It consists of long chain
Yes, sir.
of alpha glucose in helical form,
Yes, sir.
right? And it is not straight. All these alpha glucose have 1,4 glycosidic bond. Fine. So we can differentiate it by having my in mind that amylopectin.
Yes, sir.
How we can differentiate it? It
Bond.
is a monomer of alpha glucose, is a branch chain, right? And here you have, in case of amylopectin, like
Yes.
you can observe, like it would be a structure that is like this. I can show you, like it is a branch chain. Like this is how these glucose and other structures will be involved here. Okay,
Okay, sir.
like this, like branch, just like tree, that you have a tree in a very similar way. This structure is distributed. So in case of amylose, you would have like this structure. You would have structure like this. Fine.
Yes, sir.
So one is uh, helical structure of amylose. On the other
Okay.
side, you have the branch chain. Here is a helical structure. So you can differentiate these two structures. Both are macromolecules.
Yes, sir.
Both contain the same alpha glucose, right? But
Okay,
they
sir.
have just a difference. One is different in a way that one is helical and another one is branched. This one is helical, meaning amylose is helical, and the
Sir.
amylopectin is branched, right? That's the main difference between these two
Okay, sir.
structures. And one thing you must keep in mind, in case of amylose, you
Yes.
have one four. That's a difference. Okay. Here,
Yes.
in case of amylose, we have 1,4 glycosidic linkage,
Yes, sir.
right? While in case of amylopectin, that is a branch chain. So here you have 1 4 and 1 6. Okay, 1 4 and 1 6 glycosidic chain. It is
Okay.
less branched compared to the glycogen. So we will discuss the glycogen as well. So in case of glycogen, you will have a dense branching there. Number of branches would be present in case of glycogen, but here in case of
Yes, sir.
amylopectin, branches are present, but they are very less in number as compared to glycogen, right?
Okay.
So because it is a branch, so here you have number of symmetries like 1 4 1 6. So these specifically
Yes, sir.
two arrangements are present in case of amylopectin. Got it?
Yes, sir.
So by the combination of these amylose and amylopectin, there will be a structure that is what we called hmm, starch. Okay,
Starch.
so starch is actually the combination of amylose and amylopectin. Both are macromolecules, both are polymers actually, polysaccharides, not polymers, both are polysaccharides.
Yes, sir.
So these polysaccharides are further combined with one another by the combination 1 4 and 1 6 arrangement, and they form a starch, because starch is a very complex structure. Starch is very complex polysaccharide. So
Yes,
here we
sir.
have the number of arrangements. Okay,
Yes, sir.
you can also add here one more, that is glycogen. It is also a polysaccharide. At least we should complete this as well if we have time. So glycogen.
Yes, sir.
Uh, Arrays, things are clear up to now. I mean,
Yes, sir.
any difficulty, so you can discuss.
No, sir.
Okay, fine.
सर
So
अमाइलोज
these
और अमाइलोपेक्टिन का ये जो
okay.
मोनोसेक्राइड का स्ट्रक्चर होता है कंबाइन करके, ये तो नहीं होता बनाना?
Uh, in case of amylose and amylopectin, no,
यस
no,
सर.
you don't need that. Just you should know about one thing, that if the arrangement is there, like in case of, you should know the difference. Amylose is 1 4 glycosidic bond, just like the glycosidic bonds that we have discussed, right?
यस.
Here it's a mixture, like we have discussed 1 4 and 1 2. So
यस सर.
but in this case, we, another additional thing is like you have 1 6 arrangement as well, right? So that is not difficult,
यस सर.
just you should know where is carbon number 6 and where is carbon number 1. So when you combine them by the process of condensation, you will get
यस.
the amylopectin. So
एक्टिव.
there you have this difference, right? In case of glycogen, as we know, what is glycogen? Once it is a term that always glycogen. It is very familiar term that we should know about it. Glycogen is a stored form of carbohydrates.
यस.
Stored
फॉर्म ऑफ.
form of carbohydrates, right?
यस सर.
If you have eaten carbohydrates, and if your if your body is consuming carbohydrates, and it is possible that the requirement of the carbohydrates to your body is different, like you may utilize less number of carbohydrates as compared to your intake, right? So the extra amount of carbohydrate will be stored in our body, right? For example, uh, we will observe this, like it is the process, mostly it is carried out in liver. So there this carbohydrate is normally it is stored, and as a result, once you need that, for example, you are not eating something, you don't have time enough to go and take some carbohydrates. When your body will feel that you have less number of carbohydrates, so this glycogen will be degraded or decomposed and converted back into the glucose,
यस
just
सर.
in order to provide you energy, right? So it is just a storage form of carbohydrates. When you need it in the body, so they will be decomposed because it is the polysaccharide. Once it is decomposed or degraded, it will be converted into a monomer, that is a glucose. And you know very well the glucose
Yes.
is the simplest form of carbohydrates, and our body actually extracts energy from the glucose, right? Not from polysaccharides, not from disaccharides. They have to be degraded and decomposed back into the simpler sugar. Even in our body, what we eat commonly in the form of carbohydrates, till the small intestine, that carbohydrate is degraded and decomposed and transform into a simplest sugar, that is glucose. Only glucose can be absorbed by our blood. Our blood capillaries can only absorb the glucose because that is the simplest sugar, right? Not
Yes,
to
sir.
polysaccharides and not to higher sugar. Are you getting this point?
Yes, sir.
Okay. Monomer, again, when we will discuss the polysaccharides, we will also talk about the monomers. What type of monomer is here? So monomer. So here in this case, again, you would have alpha glucose, a glucose, right? Oh, okay.
सर ये अल्फा बीटा का डिफरेंस तो बता दें जरा.
Okay. Alpha beta, it is the difference of symmetry. It is
सर
the difference of.
वही है ना कार्बन वन में जिसका एक एच ऊपर होता है और ओ एच दूसरे का नीचे, ये वही है ना.
Mhm. Okay. All right. All right. Any other difference?
मुझे तो सर जी अभी यही याद है थोड़ा सा.
Mhm. All right. So commonly we always talk about its difference is like it's a difference of symmetry. Again, just like as you said, the hydrogen is, in one case you have the hydrogen on upper side and the lower side. I mean, alpha glucose molecules are actually, they are linked together to form the glycogen or starch. That is always in that we normally call them alpha glucose, right? Because the symmetry is there uh, on the. If we are talking about beta glucose molecules, they are always linked together to form the cellulose. We will discuss them. It is just the difference of symmetry. As uh, up till now, the glucose that we have discussed, they were all alpha
यस.
glucose normally, right? But
यस.
when we will discuss the cellulose there, so we will also discuss this difference, the how the glucose of uh, starch or other polymers or polysaccharides, how that is different from this cellulose, right?
यस
Till
सर.
now you should know the whatever the glucose that we have discussed, they all were alpha glucose, right?
यस सर.
So difference is only the difference of symmetry, and definitely we will discuss as well when we will talk about the cellulose, right?
यस सर.
So alpha glucose is here, that is, it is highly branched structure, as we have discussed in case of starch. You have discussed that their amylopectin is branched, but that is less branched as compared to glycogen. In case of glycogen, this alpha glucose is highly branched, right? It is a dense branch structure. Like you would have this, like for example, like this. There would be a lot of branches this way and this way, like this. Okay, so
यस सर.
number of branches are here, dense branch as compared to starch. So this is glycogen actually. This is glycogen. So
ग्लाइकोजन.
this glucose is combined again. Yes, it is a glycogen, because we are discussing about glycogen.
यस सर.
So what I am talking about, this alpha glucose is repeated enough that it is forming a glycogen, but that glycogen is densely branched as compared to the amylopectin of the starch, right?
यस. यस सर.
Okay, so this is one thing. And there branch structures with more terminal ends, number of terminal ends, like as I have shown you that number of terminals. It is compact because the branches are actually producing a compact structure. So it is a compact structure, right? Not soluble. Remember, it is not soluble. Not soluble.
Yes, sir.
Okay. So
Yes,
it contains again it contains one four and one
acidic,
six
like acidic bond.
glycosidic linkages.
Yes, sir.
Glycosidic
Like,
linkages. Are you getting this point?
yes, sir.
So where it is stored, I have told you where it is stored. It is stored mainly it is stored in
Okay, sir.
liver cells, and it is also stored in muscle
Okay, sir.
cells, right? So these are the two main cells in the body in the animals where this glycogen is stored because it is the storage form of glucose, right? The extra amount of glucose is converted into the glycogen and that is stored in our body. So once we need it back, so this will be converted back into the glucose to provide energy in our body. Okay.
Yes, sir.
So this is the point of related to glycogen. So I think we should conclude here. So next in upcoming class we can complete this topic, uh, and we will discuss two more topics, that is lipids and uh, proteins, right? So these are important chapters having some concepts with it, so you should understand these well.
Yes, sir.
So otherwise all other chapters are not enough difficult. So it is also not difficult, but just here you have some structures that you need to differentiate those structures. At least you should know what is the difference in glucose, fructose. At least you should know the differences in monomers, galactose. So when you will have the difference in their structure, then it would be easy for you to understand it, how they form the glycosidic lipids. Okay,
Yes, sir.
that's all for is. I think we should conclude it.
Yes,
So
sir.
inshallah, see you tomorrow in upcoming class. I mean on Monday.
Yes, sir.
So we will try hopefully till first or second week of March. I think I'm busy, so then we will move. Uh, Maybe to open our Saturday as well. No issue. It depends on you, uh, that how we
Mm,
can work on it. Right? It depends
yeah.
on the syllabus that how much we can cover. Okay.
Yes, sir.
So that's all. Goodbye as well, and recording stop
सर
कर
वो
दें.
मैं वो मैं आपको इंशाल्लाह आज कल कर दूँ कल कल
Okay,
तक मैं इंशाल्लाह कर दूँ ये क्लासेस आज ले लूँ मैं रात को कंप्रेस
oh,
करके कल आपको भेज दूंगा
okay, thank you very much. Okay, see you inshallah Monday. Okay, Allah
इंशाल्लाह
Hafiz.
इंशाल्लाह अल्लाह हाफ़िज़